It is truly our pleasure this evening to bring you Dr. Siddhartha Mukherjee, a cancer specialist and researcher who is on the forefront of discovering new treatments using innovative biological methods. A Pulitzer Prize-winning author, Dr. Mukherjee has been equally energetic in writing the story, if you will, of cancer, a disease that sickens and kills millions of people around the globe each year. Tonight, he will be talking about his newest book entitled The Gene, An Intimate History, which debuted at #1 on the New York Times bestseller list. We have asked Roy Jensen to introduce tonight's speaker. And in my conversation with him earlier, he just wanted me to say, "Here's Roy," and let him come out. But I think he warrants a bit more of an introduction than that. Dr. Jensen was born, that's a true story, by the way, was born in Kansas City, Kansas, and he earned his bachelor's degree in biology and chemistry from Pittsburgh State University. He graduated from Vanderbilt University School of Medicine, where he completed his residency in anatomic pathology and a fellowship in surgical pathology. He subsequently was appointed assistant professor in the departments of pathology and cell biology, later earning promotion to associate professor. In 2004, Dr. Jensen returned home to Kansas, where he was named the William R. Jewell, MD, Distinguished Kansas Masonic Professor, Director of the University of Kansas Cancer Center, Director of the Kansas Masonic Research Institute, and Professor of Pathology and Laboratory Medicine, Anatomy and Cell Biology, Cancer Biology, and Molecular Biosciences. He currently has over 150 scientific publications to his name, and he has lectured widely on the clinical aspects of breast cancer pathology. Since becoming director of the University of Kansas Cancer Center, Dr. Jensen has recruited and led a truly world-class team, which resulted in the center achieving its status as a National Cancer Institute designated cancer center, and I can't think of a better person to deliver this introduction to our speaker. So please help me to welcome Roy Jensen.
Well, thank you very much, Clarence. It is my pleasure to introduce our speaker tonight. And when Victor asked me to do this, I was absolutely delighted for a couple of reasons, one of which is I was really happy to be able to come to an event for the Hall Center for the Humanities. It is a critical part of our cultural community here in Lawrence and really across all of Kansas City. But also, the other reason is that I have heard Sid Mukherjee speak on several occasions. And you are truly in for a treat tonight. In addition to being a world-class cancer clinician and researcher, and Pulitzer Prize winning author, he is an incredible storyteller. And he has chosen to devote that aspect of his life to really talking about the intersection of science and society and how that collision impacts all of us. And at the reception before this lecture, he was telling us that this latest book that he's going to be discussing tonight is going to be part of a 3-part series. And I won't steal any more of his thunder than that, but I was delighted to hear that he's going to be continuing in this vein of really helping to illuminate some of the most important issues that we face today as a society. So without further ado, I'm delighted to introduce to you Dr. Siddhartha Mukherjee from Columbia University.
Thank you for that very warm introduction. And thank you everyone for coming. Many of you might have expected an abstract talk about medicine and science and the future, which I will certainly try to deliver. But let me begin by asking you to just have a quick look at your neighbors. Just acknowledge who is around you, because I'm going to ask you, in all seriousness, I'm going to ask you 2 or 3 questions. And I want you to think about them. You're welcome to answer them. Raise your hands if you want to. If you want to abstain, that's fine. How many of you want to know the height of your unborn child? The future height of your unborn child. You don't know your child is about to be born, and maybe in 20 years from now, a genetic tester, counselor, a private company says that with a great degree of accuracy, 60, 70, 80%, I can predict for you the height of your unborn child. How many of you would sign up for such a test? Raise your hands. Look around. Okay. Question 2. You have 2 children, and far from being able to predict with that 60, 70, 80% accuracy, we now drop the odds to 5 or 10%, but the consequences are quite serious. So how many of you would choose to know if your child carries a 5%, say, risk of a severe psychiatric illness in the distant future? And maybe we know that there are no treatments right now, but 10 years from now there might be new treatments for this. Raise your hands. Look around. Okay, these are uncomfortable questions, I'm sorry, but they're important questions. Question 3. 20 years from now, our society has changed fundamentally. We routinely have in vitro fertilization. And in that process, we sequence the genes of these pre-implanted embryos before they're implanted. Are you comfortable with a world in which we sequence these embryos, and would you be okay with the idea that we eliminate an embryo, say, where we detect a 5 or 10% chance of a future risk of cancer, and that's the only thing you screen for? You don't know whether this embryo happens to be the one that will be productive, the President of the United States, a creative genius, etc. Are you comfortable with a society where we screen such embryos and decide only to implant embryos where we know that the risk of cancer is diminished from 5% to 1%? Who's comfortable with this society? You are, yeah? Several people are. Raise your hand, be proud of it. Own the idea, yeah? A couple of hands here. Okay, so I'm going to talk today about, far from talking about what you might consider some abstract ideas, I'm going to talk about ideas that are very relevant to us today, because these kinds of questions are questions that we will, I think, face increasingly as physicians, as human beings, as scientists, but also, of course, in society at large. I will talk about cancer because I think the payoffs of the genetic understanding of cancer is deep and consequential. But I also want to talk about what might happen as we broaden our understanding widely outside very small realms of cancer, and talk about what it means to each one of us individually. Very widely speaking, I want to talk about genetic prediction and manipulation. I want to talk about deep learning, and I want to talk about this idea of precision medicine for cancer. What does it mean now that we begin to understand, or have begun to understand, cancer in a much more fundamental and rather precise way in individuals? And one of the things I want to show you, of course, is that these things, these 3 ideas, the idea that you can read much, much more deep information about the human genome that was previously possible, the idea that you can use computational algorithms to understand pieces of that information that were previously invisible to us, and the level of comfort that we have, or discomfort that we have, once we unleash our computational overlords on our own genomes, what that might do to our societies, what that might do to ourselves, whether these will create deeper inequalities, whether they will solve diseases, that we've been hard to solve. And I also want to talk about the rich payoffs, the idea that you can use this understanding to create new medicines in cancer and diseases, neurological diseases, heart disease, things that we've struggled to cure and treat in the last few decades. So before we do all of that, I'm going to delve into a little bit of history, tell you a little bit of a story, because this is the story of this book, of my book, but also it's the story, the larger story of the human genome. The story begins in a strange place. It doesn't begin in a laboratory. It doesn't begin in a university. It doesn't begin in a standard place that you might imagine the most consequential, and I would argue the most dangerous idea in the recent history of science came from. It begins in a monastery. It begins in a monastery. I spent time here. I spent a week and a half here 6 years ago in a small town called Brno. And that's where, in this walled garden, small area, in this room over here, this corner room overlooking the walled garden, a man named Gregor Mendel began to perform some experiments. He was a monk. He had been rejected multiple times from university education. He was a failed biologist, which should give us all a lot of reassurance that you can essentially write the most important paper in the history of biological sciences, having failed your examinations in biology. But here he was. He was a monk, and he was a substitute teacher. And he began to experiment, we think, although we're not sure, in this little plot of land which is weirdly enough planted with gardenias. But he, of course, Mendel began to plant peas. Why you would take Mendel's garden and put gardenias in it, I don't know, but anyway. But Mendel began to plant peas, and he made, in 1864, 1865, an incredibly important realization. And that realization was that heredity, the fact that you and I resemble our parents somewhat, that is carried not by some kind of miasmic substance, some kind of peculiar weirdness, but in fact is carried in a discrete form, like information moving from your parents to the offspring, from your parents to you, in a kind of discrete informational form, like words in a sentence. You can't tear those words apart, they will lose meaning. You can't scatter the letters apart, they will lose meaning. Like the word word, it is inherited in fullness from your parents to your offspring, to the children. And Mendel described all of this just by looking at, in great detail, by looking at peas, and it had many consequences. He realized that in every generation, information in this discrete form, not in the kind of blended form, is moving from one generation to the next. Now, just to contrast this with a very simple idea which existed before Mendel, people thought that your nose or your eyes are some kind of intermediate between your father's and your mother's nose and eyes. And this was human intuition for generations, that we were the products of some kind of blending, that the genetic information was taken from your father and from your mother and blended in some kind of biological blender, and your nose came out of that. It's a very intuitive idea. It makes a lot of sense. Except, of course, it has one big problem. It takes you 2 seconds to figure out what that problem is, and it's called gender, right? So it couldn't possibly be true that in every generation there's some kind of blending going on, because otherwise our anatomies would become genderless. So there has to be a reason that you and I are inheriting information from our parents that is somehow kept generation upon generation intact. And it's astonishing that would take Mendel in 1864, several thousand years after many of the fundamental tenets of biology had been discovered, to essentially rediscover this idea that, of course, that information, hereditary information, the thing that makes us, that gives us our anatomy, our physiology, our traits, our and by extension our identities, is not being blended in some kind of biological blender, but is carried out in discrete, in this form that reproduces itself generation upon generation. And that's the only way you can explain how gender, male, female, remain anatomically, physiologically, remain intact, as it were, generation upon generation. There's no other simple explanation for that. What's interesting to me about that story, about the story of the discovery of genes as a fundamental unit, is that soon after Mendel had discovered this, biologists who worked on genes in the early 1900s, we're speeding forward a little bit, in the early 1900s began to realize that this was a powerful and dangerous idea long before they knew what it was. What carried this information? How was this information transmitted? What was its relevance to diseases like cancer or metabolic diseases or diabetes? What was its relevance of this information that moved from parent to offspring? Long before we knew all of this, the movement to use that information to discriminate between human beings, to identify those who were weak versus those who were strong, already was being practiced. Here are a couple of examples of that. It's interesting that the most powerful examples of what we call eugenics, the idea of using genes to understand and thereby the hope to create a better society by using genetics, was actually born very soon after Mendel. It was born in the UK, it was born in England, and led really by a man who coined the word eugenics itself, Francis Galton. Galton was a cousin of Darwin's. And while Darwin was thinking about evolution, Galton was thinking about social evolution. How can we make better societies? And of course, one way to make a better society, he thought, was by selectively breeding the best together, by selectively breeding the people who were the best adapted, the best looking, the brightest, the smartest. These were Galton's own ideas. So this was so-called positive eugenics, the idea that you would use this feature, the selective breeding of human beings, to create better humans, better societies, better races, better countries. The idea then morphs and comes to the United States. Now recall that we haven't even spoken about Germany yet. In the 1910s and 1920s, where these kinds of advertisements are popular. You would open up a magazine and see something like this. And in the United States, it morphs into a kind of practical realm. In the United States, it moves from selective breeding to selective sterilization. Why don't we sterilize women or men, but of course women predominantly, who are likely to carry defective traits into the next generation and thereby remove their influence from future societies. This is Carrie Buck. My book is dedicated to Carrie Buck. I would encourage everyone to read that history. The history is very pertinent to this state as well. Carrie Buck was one of the first women to be sterilized under this law of trying to make sure that the best, that people who were unfit to breed were sterilized. The case climbed to the Supreme Court, and there was a judgment passed down, which is very chilling. It says 3 generations of imbeciles is enough, and Carrie Buck was forcibly sterilized to prevent the birth of what was then considered genetically defective, mentally ill children. It turns out, in retrospect, that in fact Carrie Buck had really no illness. She was just one of us. But it's a reminder of how quickly, from 1864 to 1920, barely a flash of time, that the idea of heredity, of genetics, of inherency was being moved from an abstraction, something that someone was planting, a monk was planting in peas, to an idea that was used as a mechanism of social and biological control. Here, of course, is that example of that. It is only after all of this, and remember, we still don't know what the carrier of this information is, Who's carrying this information, where this information comes from? Is it a chemical? Is it a substance? What is it? We don't know any of this. We're in the 1920s, 1930s, that same idea morphs from eugenics as a mechanism of breeding the best and the brightest, as Galton would have it, in England, to eugenics as a sterilization effort against women who carried what was thought to be genes of imbecility and other weaknesses. And ultimately, it morphs to its most macabre form, which is the form that we remember it today, which is Nazi Germany. Here's a Nazi physician measuring twins. Here is actually one of the most interesting images that I found while working on the book. SS cadets, you can see a swastika in this man's hand, watching on the blackboard. And if you look in the blackboard, the man who is the captain of the Nazi party has Mendel's laws and Mendel's diagrams up on the blackboard. Here is, from the United States, a Better Babies contest. Some of you might remember these Better Babies contests. Here are babies being judged for being the most eugenically fit. They are being judged for the idea that you would create a future race where this would now become racially cleansed and therefore superior as a mechanism of social control. Barely, as I said, 40 years after the discovery of the idea of the gene itself. One of the consequences of the war was that it brought a sense of shame to this project, a very important sense of shame to this project, an embarrassment because of the Nazi excess of it. But one of the things I want to talk about is that it never fully decapitated the desire to improve ourselves, the desire to improve our societies, the desire to improve our own children through control of heredity is an ancient desire. And that's why I asked you those first few questions, to ask ourselves about what we really desire. What do we desire? What kinds of things are we going to allow our societies to move into as we move forward into understanding more and more about ourselves? Keep that thought in mind. So I'm going to go back into the history. I'm going to bring you up to quickly. I'm going to bring you up to where we are today, and then go back into these questions that I had just asked you before. So the explicit eugenic desires of the 1920s and 1930s were contained, partly because of the excesses of what happened in Germany in the 1930s and 1940s, the extermination of human beings based on eugenics. The progression from better breeding to sterilization to extermination was of course a step way into the abyss of eugenics. And by the 1940s, we had decided as societies to keep aside those questions, move them away, and instead concentrate on what was it that was transmitting this information across generations. And that, of course, turned out to be a molecule that was very unknown to most biologists, called DNA. If you said the word DNA in the 1920s and 1930s, you said you were working on DNA, people would say, what a weird thing to work on. It is a strange molecule, not interesting, probably some structural component of the human body. Who would have thought that this would be the carrier of information? But in fact, this is a picture of Rosalind Franklin, whose birthday we celebrated a few months ago, looking down a microscope and taking photographs of the substance that she and others by that time had known and had established was in fact the molecule that Mendel had identified. Mendel didn't know any about the molecules, but the substance that lay behind Mendel's idea that heredity was transmitted across generations. And the final structure of that molecule was solved in the 1950s by Jim Watson and Francis Crick. And here is that first model of that molecule, a double helix. All of us every cell in our bodies contains this double helix. If this double helix was in a single cell, was expanded out, it would be, and put all together in a long chain, it would be several meters long. And each cell of our body contains this information, this twisted double helix, and condenses it many-fold, almost a million-fold, into that cell. And that information tells the cell how to behave, what to be, how to act, and it gets transmitted from us to our children through sperm and eggs. And that is, of course, the secret behind the transmission of information from one generation to the next. Now, if I took your genome, it would contain this double helix, obviously in this double helix form, this chemical form. It would contain 3 billion letters of DNA, written ACTG. And if you think about that, if it was standardly printed on an encyclopedia, it would be about 70 full sets of the Encyclopedia Britannica, one human being's genome. So all the walls, you can imagine, in this room would be lined with your genome. And if I wanted to move to your genome, I would have to make another auditorium this size and line all the walls again with the encyclopedia of your genome and so forth. So each one of us carries within our bodies one such 70-set Encyclopedia Britannica. Now, we knew this, we began to understand this, and we also began to understand that you could pick out one volume, one page, and it would be inscrutable to us in the 1960s, 1950s. It would read ACTG, CCTG, and so forth. We would have no meaning behind all of this. But how does this encyclopedia specify us? How does it specify us in the structure of our body, our forms, ourselves? And does it also specify things that are beyond simple, not just the shape of your nose, not just the color of your eyes, but of course the propensity to develop diseases? Does it specify fate? Does it specify the capacity to develop a mental illness? Does it specify our choices? Does it specify identity? How much of identity? How does it interact with the environment, this code that each of us carries, and make us? These are the kinds of questions that we began to think about in the 1960s and 1970s. We didn't know the answers to. And as we understand the answers to these questions, on one hand, we acquire the capacity to cure and deliver promises for incredibly important diseases. I'll talk about that. But also it opens up deep questions about who we are and what we need to know and how much we are willing to know about the future. But back then to the question of, okay, so you have this encyclopedia. Each one of us has an encyclopedia. How does it make us? How does that encyclopedia, which is a kind of information, ACTG, CCT, CCT, so on, for 3 billion letters, how does that make human beings? Well, the simple answer is that it makes human beings, it makes animals, organisms, by creating messages, and those messages ultimately become functional. Those messages become actualized. The difference between Sidney Brenner and Jim Watson, the difference between younger Jim Watson and an older Jim Watson, etc., are because the genome, there are 2 different genomes in 2 different cases. They have interacted with the environment, with 2 different environments. They've specified functions, and these 2 functions have ultimately translated themselves into 2 very different people. And there were molecular correspondence to this. You don't need to know all the details, but the point is that the gene was carried in DNA, as I just said. This message is RNA, and the form and function is a mixture really of RNA and protein, but really is this piece of it. The scientific details are unimportant. The most important piece is that somehow or the other, for most purposes, this Encyclopedia Britannica, 70 volumes of it, gets that message, the information carried in it, much like a computer gets translated into real words, real information, and then gets actualized in our bodies. And work done by several other people. This is work done by Marshall Nierenberg. We learned to read how this information, we began to learn to read how this information actually gets actualized. And by the 1950s and 1960s, again, this idea that DNA makes a message, makes a protein, was made obvious. There's one last thing we need to know, and that's important, and that is that this is a dynamic process. Here's the best example of the dynamism of this process. This creature, which looks nothing like this creature, both of these have the same DNA. Their information is exactly identical. So somehow or the other, in this state, the genome of this caterpillar reads out caterpillar, and within the matter of several days, several hours, that same genome is read out to make a butterfly. And if you understand this, you begin to understand how you can have thousands of different cell types in your body, but every cell carries its own genome. Your liver cell, your brain cells, your nerve cells, your blood cells all carry the same genome. They interact, this genome sort of expresses itself. This encyclopedia makes different versions of itself. It actualizes itself differently in different cells and different organs, and that is called gene regulation. So by the 1950s and 1960s, we had realized all of this, that there was a master encyclopedia that we carried in ourselves. This master encyclopedia allowed the creation of very different forms by reading out subparts of it. But in fact, despite that, the master message, the encyclopedia, remained the same. But the question was, there were two questions. Could we read the actual letters? And number two, could we change the actual letters? And that's what I'm going to spend the rest of my talk thinking about. What if we learned to read all of this information? And what if we began to write some of this information? The reading of the information began in the 1970s when Fred Sanger deduced some mechanism. You don't, again, need to know about the details, but to really decipher what the actual code was. So all of a sudden, we began to read A, C, T, G, C, C. We began to understand what the actual manifestation of the code was. So you could say, what is the code on page 370 of your encyclopedia? It was unknown to us. We knew there was a code. It was unknown to us, but we began to read that code. So I call that reading genetic information. And the second part of that was that we began to find mechanisms in science to change genetic information, to combine it from one to the other. I'll give you the simplest example. What if I took your encyclopedia, took page 370, and swapped it with someone else's? Move those two pieces around. It would be like writing a new volume of a new encyclopedia, correct? So by the 1970s, we had just begun to do this. We had just begun to find ways to sequence or read the human genome, and we had just begun to create ways to swap the human genome. And this already had many, many consequences. I'll give you the simplest consequences. Take a medicine like insulin. A medicine like insulin, it's very hard to extract, but what if you could take human insulin, tear that page out of the human encyclopedia, and put it into a bacteria and make the bacteria make human insulin, or make a yeast or some other cell make human insulin. All of a sudden, you've just essentially swapped one gene from one organism to another. You can move genes across the biological kingdom, and you begin to invent a whole universe of new medicines because you've torn out one piece of the encyclopedia, moved it to another animal, moved it to another plant, and begin to manufacture it. In fact, this is work done by Herb Boyer. Bob Swanson was a venture capitalist, and here they are actually showing how you can, on a blackboard, they're showing how they started thinking about making drugs like insulin. Obviously, this reading and writing must create fears about who we are as human beings. This is a comic strip that I found from the 1970s, in which the fear is, what would happen if we started swapping this information without control. The idea of the evil brotherhood of mutants. What would happen if you started moving information from one organism to another organism, making, not by natural means, nature makes mutants all the time, but making mutants by artificial means, by tearing out words, moving words from one encyclopedia to another. And what would happen when you began to understand human diversity in terms of these kinds of methods? What would happen when you started asking the question, what's the difference between this human being who is obviously much shorter than the rest of us, and this human being who's a physician who carries genes that make him or her much taller than these patients that he or she is treating? What would happen between those differences? What if we could read those kinds of ideas? That's why I asked you a question. How many of you would like to know about the height of your future child? And this reading and writing of information, this is my mentor, Paul Berg, who was one of the first people in medicine to start swapping genetic information between organisms. He invented techniques to say, take a gene from a human being and move it into a virus, or take a gene from a virus and move it into a bacteria. And the fears that that created, this is Fred Sanger, so I call this writing, I call this reading. The fears this created, of course, went right back to the great fears of the 1920s, which is the fears of eugenics. What if we took this information, used it incorrectly? Would we start worrying about creating the so-called perfect race? So this is where we sat at the edge of reading and writing in the 2000s. And then we moved over that edge. We went over the edge because we really started reading and writing human genes in a way we hadn't before. This is the draft sequence of the human genome published in 2001. The cover of a magazine, the Science magazine, was the sequence of the human genome, the draft sequence of the human genome. The first fully read sequence. Obviously, there's some pieces that are still missing, but the first fully read sequence, near fully read sequence of the human genome. I find it very interesting that all the people in this picture, none of them have disease, or at least obviously disease. They're all examples of human beings. This guy looks like George Clooney, by the way. But all of them all of them, of course, signal the idea of normalcy. And this was the draft sequence of the human genome. This was the beginning of our reading revolution. So what did we learn from all this reading? We learned a lot. Number one, we learned that most human traits are controlled by multiple genes. Very important thing to realize. Your nose, your eyes, the color of your hair, your height, aspects of who you are controlled by not one gene, typically, but by multiple genes. There are exceptions to this, obviously. And therefore, as a consequence, most common diseases are polygenic. By polygenic, we mean multiple genes. So heart disease, cancer, most common diseases that we encounter, aside from the rare diseases, are controlled by multiple genes. We should know all of these facts, actually. These are going to be principles that allow us to tackle the questions that I asked you to start with. Many diseases have genetic links, but right now, we're still in the reading phase. We still don't know the exact genes for most diseases, and we certainly don't know most genes that govern fundamental aspects of normal human traits, height, hair color, eye color, and so forth. This is a very important equation. I think it's sort of like the equation that governs fundamental laws of physics. The equation that governs fundamental laws of biology is that genes plus environments plus chance gives rise to traits. Most human traits, and we really need to understand this, most human traits are not governed entirely by genes, are not governed entirely by chance, but by a combination of this. In contrast, some things are highly genetic, so in some traits, genes will dominate. In some traits, environments will dominate. and occasionally chance will dominate and determine what our ultimate traits are. But we can ask this question feature by feature, disease by disease, trait by trait. So in other words, if someone asks me the question, is it nature or nurture? Is it gene or environment or chance? I immediately ask them back the question, well, what are we talking about? Just to give you, again, classic examples, most of gender anatomy and physiology has a genetic component, whether you are born in Antarctica or whether you're born in Tanzania. Fundamental features of male and female biology, for the most part, will be governed by genetic components, the anatomy and physiology. There are other aspects of gender identity, features, human identity that will be governed by some combination of genes plus environment plus chance. There are aspects of diseases that will be governed by environments and chance. And this will go on over and over again. So whenever people ask me the question, well, is it nature or nurture? I always ask back, well, what feature are we talking about? Because it's going to differ from feature to feature. And we can identify, it's not a null question, but it's a question that's very dependent on what we're asking about. And as a consequence of all of this, if we were to imagine a genetic report card, this gets back to the first question that I asked in the beginning. What if I gave you a report card that said, you know, you had a 5% propensity to develop a lethal disease or a terrifying psychiatric illness? That genetic report card will usually be given in the form of propensities. It won't be A plus B minus C plus. It'll be, you'll have a 20% chance, a 10% chance, a 5% chance to develop breast cancer in the future, a 12% chance to become, to manifest X or Y feature in your future. What's interesting about all of this is that all of this is true. Most human traits are controlled by multiple genes. Most common diseases are polygenic. Most have genetic links, but the exact genes are still unknown. That genes plus environments plus chance gives rise to features, and that we generally will give rise to only propensities. But what's interesting about this is that our computational power, our understanding of where genes is going, for some features is reaching the place where we can actually start predicting. And here is exactly the question I asked you before. This is something that came out 2 weeks ago, and it took me by surprise. This is a genetic predictor of human height based on genes alone. So here is the actual height of a human being, and this is the predicted height of a human being. And obviously, if it was exactly correct, it would be a straight line up like this. These are about 2,000 human beings. What's shocking to me is this curve doesn't look like a mess. It looks pretty good. And for statisticians here, it's not great. This R value is 0.64. You could have some arguments with it. Obviously, there are people that are off the straight line, but it's not bad either. And that's what's shocking to me, at least. In other words, in this experiment, they knew nothing about a human being except for the sequence of their genome. And based on that sequence alone, they predicted one feature that we find pretty complex, human height. And they could predict that just by reading that encyclopedia. They read it. The encyclopedia was predictive of human height. Why did this matter? It matters because it has consequence for virtually every human disease. It matters because we are trying to understand maintenance and risk. It also matters because we really need to figure out how to deal with things like insurance. What if we were to predict our own risk in the future? How would we insure anyone? How on earth could we insure anyone if we began to understand what the risks were in the future? So that part is about reading, what happens when you begin to read. But in the midst of all of this, we're also beginning to write. And by write, I mean, remember I said you can change the actual content of the encyclopedia. I've called this loosely writing the human genome. And these are early days yet, but these are some scientists. This is Jennifer Doudna. How many of you have heard of the word CRISPR? So virtually everyone. This word was obviously not even heard about 10 years ago. Jennifer is one of the inventors of CRISPR. She's a colleague. We actually, my own lab works on CRISPR a little bit. I'm doing a talk with Jennifer in January. But in brief, again, now we have the analogies intact. Here is a mutation that causes breast cancer, and here's a schematic in which you could go into the encyclopedia and potentially in some cell, maybe an embryonic cell, maybe the cell of an embryo, you could correct that mutation. You could erase that. Go to page 375 in that encyclopedia, go to page 375, paragraph 4, and where it says CCTGC, make it CCAGC, or something like that. And on one hand, you and I can appreciate how powerful that would be to eliminate terrifying diseases or terrifying risks of terrifying diseases, breast cancer, pancreatic cancer, neurodegenerative diseases, Huntington's disease. But what if we just extended that concern outside and began to think about changing fundamental features of human identity? What about someone who wanted to have a very tall child? Should we allow that, or should we say no? And so, in other words, a combination of embryology and gene editing techniques have really pushed us to a point of time where we really have to take these questions quite seriously. How should we allow ourselves to intervene on the thing that we consider most human of all, or one of the things we consider most human of all, which is the genome? Here's a slide that I find fascinating. This is from mouse cells, but very important, because here are the mouse cells. These are the cells you can genetically modify, and of course, they're being coaxed and developed to form these cells, and this can be done with human cells. And these cells, anyone know what these cells are? These are sperm. So this is a cell. You can modify this one. You can edit its genome. You can change, like I showed you here. You can erase, you can read, you can write, and you can now use those cells to make the very cells that form the basis of our of the next generation of human beings. And therefore, you could do things like the questions I had asked you before. What if you read, what if you created, what if one created, not one embryo, but several dozens, and sequenced all of them, potentially changed some of them, and you had the capacity to selectively implant only the ones that you wanted to change, or you had changed already, so-called pre-implantation genetic diagnosis. These questions have not fallen on flat ears. They have been taken up at the highest levels of concern in the United States. A very large committee met several months ago to ask the question, well, what should be allowed? What should not be allowed? Where should we stop this kind of intervention? And this was a report produced, but it'll be produced again and yet again as we move forward. So this is the report. I want you to read this, not the whole thing. I'm going to highlight some sections. But what are the concerns? Why are we worried about all of this? Well, one concern is that we will diminish the dignity of humans, that we will diminish the respect for their variety. I like the word their. It's our variety. And finally, that there's a lack of humility. We will be unhumble in the prospect of being able to change our own genetic information. Have we started actually doing this stuff on human embryos? The answer is yes, we have. We have started. There have been 3 to 4 attempts at altering genes in human embryos. The safety and on-target effects remain unknown. But here's the important part. Nothing in principle stops the field from moving forward. And there will be inevitable clashes to come. And so the committee decided there were going to be 3 criteria to develop, to allow human genetic. This is serious stuff, guys. So 3 criteria. This is kinds of technology that makes, I don't know, Uber and Facebook seem like child's play, because we are intervening on things that we hold stewardship of. And everyone here needs to know the vocabulary, because we will have to make decisions about what's permissible and not. So 3 criteria for manipulations of human genomes was decided. One is that the disease in question have extraordinary suffering. Second, that we are confident that when you make the change in the gene, you actually are producing the disease. In other words, this is not some kind of 5% relationship. There's some very direct relationship between the gene of consequence and the disease. And third, that there is no other justifiable alternative and the state is not coercing you to do all of this. These seem like very reasonable criteria. The problem is it takes 2 seconds to understand that they're very subjective as well. Who defines extraordinary suffering? What is the limit? What are the where do we draw the line in the confidence that that the gene-disease interaction is tight? Is it 50%? Is it 57%? 52%? 5%? And what does it mean? What's justifiable alternative and no coercion mean when it becomes privatized, when you can make the decision? The state says, it's not my problem, it's your problem. But you now can make the decision to implant certain embryos. You then, in 20 years' time, can sequence your, the genomes of unimplanted embryos. Who is to make that decision? I'm going to spend a little bit of time on the last phase of all of this, which again has deep consequences for all of us. I'm not going to spend a lot of time on this, and that is just to emphasize that if these techniques of reading were already powerful, then you can imagine that using very deep learning algorithms, computational algorithms, computers that learn, and using them to understand genetic information would kick this information to yet another stratosphere. I have a couple of examples of this. One example I gave already, but I'll give you a couple more. Here is an example of a young man that I know who has a genetic predisposition for a melanoma, for developed melanomas. We didn't know that. There was no one knew this. But in fact, he developed one melanoma here, a second melanoma here, a third cancer here. And it turns out that there are now programs that will scan photographs of you or scan the lesions that you develop and divide them into benign versus malignant lesions. There are programs that are being developed that will scan your genome to allow us to really incisively understand the risk of cancer. You can understand what the benefits of such therapies could be. You could be pre-screened. One proposition is to use Facebook passively, so that while you sleep, if you agree, while you sleep, your own pictures are being scanned for such the development of such lesions. How many of you would be comfortable in a world where you could, by consent, give consent to the idea that while you sleep, a computational algorithm filters pictures of you on social media to find out diseases that you may have in the future? Are we comfortable with that idea? How many of you would be comfortable with the idea that your cell phone begins to pick up, in combination with your genetic predisposition, future risk for neurodegenerative diseases like Parkinson's disease, so that when you pause, when your words change, when you begin to understand what it's, that a filter begins to pick up in combination with your genetic risk, whether you in fact are at an increased risk of a neurological disease? How many of you are comfortable with this invention, which uses in combination with your genome, so in other words, your genetic, you get gene sequenced, and then in a bathtub, as you take your bath, an ultrasound device scans you for the earliest development of pancreatic cancer? Important consequences for all that we think about. And finally, this curve that I showed you of height isn't an abstract curve. This was not possible before because we didn't have computational power. This, in fact, is this study uses deep learning, and it's only through deep learning that they could get this kind of prediction of human height. So I'm going to stop at the last point here, which is that we are on the verge of making some important developments in precision medicine. The capacity to diagnose cancer earlier, the capacity to sequence your genes earlier so that you can predict risks for future diseases, the capacity to understand information that really we didn't have before. Those are the great upsides to all of these technologies. And some of you in this audience actually are already beneficiaries of these. Early detection, early detection with computational technologies, early detection through genetic technologies, BRCA1 genes, breast cancer genes that we can sequence and therefore predict your future risk of cancer. On the other hand, there's a concern that as we delve deeper and deeper into these technologies, deeper and deeper into our own genomes, that we will begin to assault things that we consider fundamentally human, that we begin to assault things that we consider private information, information that we change about ourselves. I'm going to end with a very personal anecdote. My family history, I have a history of mental illness in my family, schizophrenia and bipolar disorder. And the question, I have 2 daughters, and the question arose at some point of time that we now begin to have, it's very early days, we're beginning to have the first genetic hints of what might increase risk of these 2 diseases. And the risks are on the orders of 2% increase, 5% increase, 10% increase in risk. And the question that my wife and I sat down with the other night was, should we sequence our 2 daughters? And if both of them are very well, thank you, but what if one of them, both of them being completely well, fine, no other illnesses, what if only one of them was to have that 5% increases? Would it fundamentally change the way we imagined that child? On the other hand, would it make us more cautious, more thoughtful about the future of that child? Would it make us think twice, screen better, think deeply about that child? It's a question that will resonate, that does resonate already within society. It'll keep resonating with deeper and deeper penetrance. So I've tried here to do several things. I've tried to show you historically where we've come to, but I've also tried to show you that where we've landed up is extraordinarily complex territory. On one hand, we have the capacity to deploy technologies to make medicine precise for individuals, including genetic diagnoses for cancers, genetic diagnosis for many diseases. On the other hand, the moral questions, the ethical questions that have been raised by these technologies are deep and profound, and we don't have simple answers to them. It is an inevitable consequence that we will have to answer these questions in the next 10 odd years. And if all of us don't get into the conversation, we don't learn the vocabulary, if we don't know the science and the history, we'll be left out of the conversation. So I'm going to end here and open up the floor to questions. I'm sure there'll be a million questions, but I'm going to end here and open up the floor to questions. Thank you.
I've been reading some in the National Geographic and some other publications about young children who, having been born a girl or a boy, very deeply recognize that they are the opposite sex. Is there a genetic connection? And if so, how much or what do we know about that genetic connection?
It's a very complicated question. Thank you for asking it. The quick answer is most people, I would say most people, and I think I'm being fair here, most people in medicine and biology think that there will be a strong genetic connection. Most people in medicine would suspect that it is not one gene, but multiple genes, so a combination, as it were, of genes. Most people would suspect that it's in the future, very near in the future, we will be able to decipher what these are. I'll give you the classic example. Some of you might think this is obvious, but let me tell you what's interesting about it. The important story is going back to the anatomy and physiology of gender, so male versus female. It turns out and perhaps not surprisingly, there's only one gene that controls it. It's a gene called SRY. If you turn it off, you go in one direction, anatomically and physiologically. If you turn it on, you go in another direction. In fact, you can take animals in which you just change the SRY gene and they become, the embryos, switch like a toggle switch from male to female. So that's the top of the hierarchy. The top of the hierarchy is one gene that controls a master gene that controls the anatomy and physiology of sex. Now, obviously, gender identification is more than anatomy and physiology. It involves a kind of computational algorithm, as it were. I don't want to make this sound too abstract, but where you take all of this and ultimately bring into your brain the identification of whether you are one gender or both, or sorry, neither, or the spectrum that lies in between. That is not, we suspect that is not one gene, but multiple genes, but we also suspect that there are genetic components to this, strong genetic components to this. So I suspect you're asking a very important and obviously a pertinent question to our times. This has been politically discussed all across the board. My personal feeling, my instinct is that it's likely to be highly genetic, one. Number two is that it's very likely that we will decipher the code in the next 5 or 10 years using, I just showed you one example of a very complicated feature, height, where we've begun to decipher the code. I'll throw back the question now at you. If we deciphered that code, would you be comfortable in sequencing your unborn child to know the code?
I think my immediate answer is yes, because of the amount of confusion, isolation, just
Yes.
general extraordinary suffering that I think these people go through who are born genetically a female, say, and know very deeply inside that they are male. Because of those factors, yes, I would want to know, and I would also want to know the other genes that impact, not just the X and Y. I would want to know what other genes impact that before I would actually want somebody going into my embryo.
May I ask you a tougher question?
Mm-hmm.
Suppose there were 10 genes, 15 genes, but one of them was very strong. Would you be comfortable with technologies that in your case you would say yes, or other cases people, would you be comfortable with technologies that allowed you to change those genes?
I think I would be, so that my child could be definitely either male or definitely female.
How many people are comfortable with that idea? Restricting being...
We have enough complexities being one or the other.
Of course, and that's the point I'm trying to raise. Absolutely right. Thank you for raising this. But this is morally complex territory. But it's not as if we can put our heads in the sand like ostriches and say, "Go, take them away," because we've discovered them. We're uncovering them day by day, and we will continue to uncover them. So thank you. I'm going to move on to the next question, but thank you. Very important set of questions. The other side, sorry. At this rate, we'll never finish the evening, so.
Hi, I just want to start off by saying thank you. As a student here at KU, you coming here and speaking to us means a lot. My question for you is, how ubiquitous do you think gene editing in utero will be, say, 10 years down the lane, 20 years down the lane? And do you think that we have a society that is adequately equipped to deal with challenging some of the advances we make in science?
Well, we just illustrated some of the challenges right there in that conversation. So just to remind us, we need to rise up to the challenges because those questions somewhat have to be answered. You don't have to be answered immediately, but we need to think about them. That's one. Number two is, now how practical is gene editing in embryos? Now in full embryos, once an embryo is formed, many, many cells, it gets pretty tough because you have to basically think about correcting multiple cells, all the cells, hundreds of thousands, maybe hundreds of millions of cells that an embryo has. On the other hand, it's much more practical, for instance, in that picture that I showed you before, which is that you can edit genes in cells that form egg and sperm. And that is much, much more practical to do for lots of biological and theoretical reasons. So in fact, the possibility that we can come to the edge of creating gene-edited human beings is actually much more practical than, or much more technologically feasible than we had originally seen. There's some major challenges still, and I've listed them here. Whether these techniques are safe, whether they only correct the genes or edit the genes of the target and leave out all the rest. In other words, only page 347, word 46 is corrected, everything else is left intact. We don't know. I suspect that that's going to be the case. There'll be a few off-target effects, but most things will be on target. So the bottom line is that for most organisms, including human beings, my suspicion is that we will get to a place where technologically it'll become feasible. Yes?
Thank you. Your examples largely make clear the potential, at least, for clinical health benefit. How do you think in terms of prospects for these discoveries for population health improvement, and particularly implications for health inequities?
For what, health inequities?
Yes.
I've written about this before. In fact, obviously, one big fear, among the many big fears, one big fear is that we will divide societies into structures where some part of the population has access to these diseases, to these technologies, and thereby begins to sequentially eliminate, for all good reason, diseases in their own children, whereas another part of the population does not have access to these technologies and continues, therefore, to have susceptibility to diseases, genetic diseases. That would be, as you can imagine, a deeply consequential political event if all of a sudden not only would we become unequal for economic reasons, but we would be split into two genetic societies. This fear has been raised in literature many times, but not really fully realized, although you could say that in fact that the attempts in the 1920s were supposed to do exactly this. My own fear is that our technologies are pushing us closer and closer to that place. Within the next decade or so, I think the prospect of having quite deep prenatal genetic diagnosis will become affordable for some people, but not affordable for others. It will be privatized, and therefore I think we will begin to see these rifts drawn through our society. I'm very uncomfortable with that kind of rift. If you can imagine the poor having no access to genetic technologies that allow them to think about their children in terms of genetic susceptibilities, the rich having access, makes me extraordinarily uncomfortable. And I think most people in this audience would feel similarly uncomfortable. Yeah.
Sir, good evening. I have prostate cancer. It was discovered about 5 years ago. I had surgery, then I had radiation treatment. The PSA keeps ticking up bit by bit. I see an oncologist every 6 months to get checked out. I want to ask a question, as you have, to our audience here. I can see most people here, but those in the front might want to turn around and look at the response I get from the audience. I want to ask people here, if you feel comfortable about it, raise your hand. How many people here have cancer? Let me see your hands.
Have cancer or had cancer?
Have cancer or had cancer?
So statistically, we know that number is going to be 50 to 70% of this audience.
Good, thanks.
Hello.
Hi.
My name is McCoy, and I'm a graduate student here. And I have a question that has nothing to do with your talk.
Okay, very good. I like questions that have nothing to do with my talk.
But more to do with sort of an observation that I have made.
Yeah.
You have a passion for making science accessible, and I know that I, who is not a geneticist, much appreciate the simple language you've used. What advice would you give a budding scientist who's also trying to do the same?
So trying to make science accessible, you mean?
Yes.
Well, my own advice is, I was driven by, I'm interested in questions. I start writing books or articles or doing experiments in the laboratory when I have a question that really, really engages me. And once the question rises to that level of interest, I become obsessed with it. I start writing about it, I start thinking about it. Emperor, my first book, grew out of a very simple question. A woman asked me, she was a cancer patient, she said, why am I being treated like this? What happens to me? Why did I end up here? The gene grew out of a different question. It was the question that I just asked you. It's a very personal question. What if you had 2 daughters at risk of mental illness in your family, and you all of a sudden discovered genetic information? So in my case, I'm very question-driven, and I think that's the simplest way in what one can become. One can start thinking about communicating questions and answers to a much wider audience. Thank you. Yeah.
Hi, I'm Julie, and I just started pharmacy school. And I was wondering if you had any advice for the next generation of healthcare providers about being a good doctor in the face of these technologies in preventative healthcare.
Well, I think the point of my talk today was that many of you, I don't know how many of you were aware, these questions, the question that I've asked, the question that I've raised, some of the issues I've raised, must have been burning in the background of everyone's minds, right? Is that true or not true? They must have been in some slow level of sort of the back burner of humankind, the filaments that drive the back burners of your questions when you're driving across vast stretches of Kansas must have been sitting in the back of your mind. My point here is to just bring them right up front, because I think they've already become important, they will continue to be important, and they will drive some of the fundamental observations and concerns of the next generation. So again, to go back to your question, there's no way that a young scientist, a young pharmacologist, a young physician can engage with these technologies without bringing these things that have been sitting in the back burner, as it were, right up front and really confronting them. What does it mean to acquire information which we didn't have before? What does it mean for equality? What does it mean for insurance? What does it mean for healthcare? What profound things can it do to our health? Let me not sell these technologies short. The capacity to annotate cancer genetically and try to get precise medicines for your cancer, which is not the medicine for someone else's cancer. What a wonderful idea. The prospect to not have a child who has Huntington's disease and therefore might suffer might be a wonderful idea. But on the other hand, just lying next to those ideas, those technologies, those events are things that obviously are concerning and quite scary. So I wanted to pinpoint those as well. Maybe we'll take a couple more questions and then stop. Last 2. Sorry. We could go on the entire night. I know everyone. This is one of the interesting things about giving this talk, I have to say, is that it becomes very quickly a talk about all of us. It involves all of us. It involves all of our individual lives. So it becomes very quickly not an abstract. It becomes about things that are very, very close. Sorry, go ahead.
Thank you for a very nice talk. I learned a lot. My question, if you remove a gene or editing a gene, can it affect the other genes?
So it's an important question, and so this is what I was trying to talk about right here, actually. So we now are trying to understand what the effect is. Number one, let's say I say to you, you have these 70-volume encyclopedia, erase, the word 16 on page 347. What would happen to all the other words? And the funny thing is we really still don't know. So this is this question of, is it on target or off target? Is it on target? Is it safe? We don't fully know for embryos, for human embryos in particular. There is a feeling, a shocking feeling, that in fact it tends to be on target. You tend not to disrupt everything else. And that goes back to Mendel to some extent, because these things are discretized. It's not like a vinyl record in which you put a scratch and the whole record won't play because it's a continuous spool of information. Because genes are quite discrete, they have their own, they move like particles or like words. you can change one word, erase the word, but the rest of the sentence remains intact. I was at a talk yesterday where we were discussing gene editing, and the example that was given is, in the past, if you took the phrase "twinkle, twinkle, little star," in the past, most of gene therapy that we could do before we invented all these techniques was a kind of crude replacement. You could say, "Twinkle, twinkle, little big star." And your cells don't fully understand that. There's a little confusion there. You have little, you have big. One of the interesting things about these new technologies now is that it really seems you can strike out the word little and replace it with big. So the cell really reads twinkle, twinkle, big star. Obviously, that's consequential. We don't know if other sentences in that phrase, in that poem, are affected or not. They may be. We'll find out. But the early information suggests that they're not affected. They're surprisingly right on the right target.
The whole reason to ask these questions, we have seen experimentally, if you insert one gene, strong gene, in a cell, exogenously, other gene gets suppressed.
This is very well known. This is very well known.
Okay, thank you.
Thank you. Yeah.
I'm sure I speak for a lot of the audience in appreciating the clarity that you brought in your presentation, and also the ethical questions that you've raised. It seems to me that there's a parallel in medical science right now with these ethical issues, and that comes from neuroscience, where we're growing capacities by detecting synapses and averting them to stop behaviors or make impossible some behaviors, such as removing anger from our repertoire of emotions. Do you agree with this parallel?
Yes, I've written about this parallel as well. One of the most interesting crisscrosses that I see, of course, is the intersection of neurodevelopmental ideas and genetics. I read several years ago, in writing the gene, I read it, and it's in the gene, because it's an interesting section. I encourage you to read it. There's an interesting section in which I discussed this article that I read that sent a kind of shiver up my spine, which is to say that, what if we could take, based on genetic information or other, maybe anatomical information, remember these things converge. You can add one to the other and you get better and better prediction. You get more and more precise prediction. What if we could take soldiers and identify those with the risk of PTSD even before we deployed them? So some people might say, well, that's a good thing. That would help us in so many thousands of ways prevent trauma. And then I began to think, do you really want to unleash a soldier who's unable to experience trauma and its consequences in a battlefield? You really want that kind of soldier? That's the worst kind of soldier to deploy. So obviously, I discussed this in the gene. I'd encourage you to read this section. But obviously, you're asking the question, what if we could eliminate certain parts of our human emotional spectrum using a combination of neurological, neuropharmacological, and genetic techniques, screen those people, give them therapies potentially up front. It is quite clear to me that universe is more morally complicated than you can imagine. Last question. Yes, sorry.
Yes, I'd like to hear your opinion on what you feel the effects of trauma are on the expression of the genetic code, anywhere from a mother who's very stressed out during her pregnancy to the more overt traumas like sexual and physical abuse.
Yeah, I think the quick answer is we don't know. And I think that we won't know for a while. So here's what we do know. You're asking two questions. One question you're asking is, when you have severe trauma of some sort, does it affect the patterns of expressions of your genes in your body? And the answer is, most people would say very likely. Your brain is being remade, you have hormones going through your body, that are changing patterns of gene expression. That's the simple part of the question. The hard part of the question is how much of those changes in genetic expression, not genes themselves, but the programs that genes are expressed in, how much of that is transmitted to your children? That's a much, much harder question, because remember, to some extent, our children, their genomes are a barrier against this kind of information penetrating into the genomes. I like the analogy between software and hardware, right? So there is a strong barrier between software and hardware in our bodies. Our experience of trauma, what we experience is our software experience. But it doesn't change the genome itself. It doesn't change the hardware of our genomes. And in fact, we don't know the extent to which it changes the hardware of our children's genomes and their children. It's a field that's being extensively studied, as you can imagine, and there's a huge amount of controversy around it. Some people say that you can pass some of this into your next generation. Some people argue that you cannot. We won't know for a while, because these are longitudinal studies. You need to study 2 to 3 human generations to know. And the only answer is that we'll have to wait to find out. So thank you very much. Really provocative and important questions. And thank you again.